Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. The phrase neurons that fire together, wire together encapsulates a key idea of Donald Hebb from the mid 20th century. When neurons that are connected fire spikes, that is, action potentials, roughly simultaneously, the synapses between them grow stronger. This phenomenon was seen in seminal electrophysiological experiments in the early 1970s, which established what is now known as long-term potentiation, or LTP. In the late 1990s, both theoretical and experimental studies demonstrated that the relative timing of the presynaptic and postsynaptic spikes plays a key role. When studying synapses between excitatory neurons, it was found that when the presynaptic spike occurs some milliseconds or tens of milliseconds before the postsynaptic spike, the synapse is strengthened. When the presynaptic spike instead occurs after the postsynaptic spike, the synapse is weakened. This phenomenon, called spike timing dependent plasticity, or STDP for short, naturally attracted a lot of attention from computational neuroscientists. And in the 2000s, numerous different mathematical STDP rules were suggested and explored. Initially, the main focus was on plasticity of synapses between excitatory neurons. However, in 2011, a seminal paper on the role of plasticity of inhibitory synapses was published. The authors were today's guest, Tim Vogels, as well as Henning Sprekler, Friedemann Senke, Claudia Klopath, and Wolfram Gelsner. This paper made an impact on me, since it suggested how certain plasticity rules for the inhibitory synapse could make a recurrently connected network of excitatory and inhibitory neurons self-tune to get to the so-called balanced state seen in many cortical networks. Before this, model parameters had to be tuned by hand to achieve the balanced state in network models. The paper also suggested that a division of labor between excitatory and inhibitory synaptic plasticity, where memories are stored in excitatory synapses, while inhibitory synapses assure that the overall network remains in a suitable state. However, it turned out to be difficult to make network models including both excitatory and inhibitory synaptic plasticity, at least if the inhibitory and excitatory plasticity rules are independent of each other. But if these plasticity rules are connected, that is, codependent, the situation is different. In a paper published last year by Everton Agnes and Tim, it was found that codependent excitatory and inhibitory plasticity could account for stable and long-lasting memories in cortical networks. And in a paper this year from Tim's group, evolutionary algorithms were used to optimize such plasticity rules. That is, to do meta-learning, or in other words, learn the learning rules. I found this whole endeavor exciting and thus invited Tim to the podcast to talk about the history of synaptic plasticity rules up to and including these last findings. As a side activity, Tim is also interested in exploring how internet may help improve the ways we do science. Together with Panos Poselos, he is running the website Worldwide Neuro, which, for example, brings seminars online and, as they say, make them greener and accessible from anywhere on the planet. A link to Worldwide Neuro can be found on the episode homepage, together with links to the papers discussed in the podcast. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. Now, I also have a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. Okay, here is Tim. So, welcome to the Theoretical Neuroscience Podcast, Tim Vogels.
Hey.
Hey.
Thanks for having me.
Yeah. This podcast is going to be about synaptic plasticity, and we're sort of, I guess, which has been a quite vibrant field, like especially this spike timing dependent plasticity for at least 25 years or something. So I guess there were some important experimental findings in the late 90s. So
Yeah,
what were they? Well, what were those?
it's an exam. Hi. So, um, in 1995, so I mean, everything's based on what wires together fires together. The
Yeah.
Carla Schatz's paraphrasing of Hebb's
Mm-hmm.
rule.
Mm-hmm.
Um, and in the late 90s, people, or I guess in the mid 90s, people started thinking very seriously about the consequences of that, and they had this computational tools to actually simulate the consequences of what fires together wires together, and um, they started thinking about whether that's the end of the story. And at the same time, experimentalists had the technical abilities to also look into this, to patch pre and post synaptic neurons, and identify synapses upon at which changes in efficacy should be happening. And I guess they realized around the same time that there was more to the story than just firing together, because the notion of together is ill defined in the original postulate.
Mm-hmm.
What is together, and so yeah, I mean, chronologically speaking, Wolfram Gersner was the first one to postulate that together meant something different when it was pre before post than when it was post before pre.
Mm-hmm.
But right after that, in 1996, there was an experimental paper and then another one that actually confirmed that suspicion that pre before post should lead to a strengthening of the synapse, and post before presynaptic activity should lead to a depression. And that was sort of, very much in line with the intuition that causal events should be causal, or should be rewarded, and acausal events shouldn't be rewarded. And so it became a very
So
popular
the
explanation.
experimental systems there was like some work done in cortex, I remember, and
I
uh,
believe so. Actually, I'll make a fool out of myself. Can I look up the paper?
no, not very, not well. Anyway, so there were like some. I think at least there was some of these layer 5 neurons in cortex or something.
Yeah, I think so.
I think Markram did some of these experiments.
It's Markram, and Markram was one of them, and then B and Poo was the other paper.
Exactly. So that's sort of like the two names I also remember offhand.
The
So
two, and they created sort of the same plot. What my lab sort of somewhat non-differentially refers to as the butterfly plot. This
okay.
increase in efficacy changes as you roll, as you approach near coincidence of pre and post, and then immediate flip to strong depression when post comes first.
The closer you are in time, the stronger the effect is, and if it's pre before post, then it's a positive, and if it's post before pre, then it's negative.
That's
Right?
right.
That sort of makes a
That's right.
nice butterfly. And then, of course, this was sort of and this led to a lot of theoretical work, right, and exploring possible effects of this. But that was mainly on focusing on this like effect of, it's like what's it called, EE plasticity from excitatory.
Yeah, I mean, the original work was done on excitatory synapses, excitatory synapses, right?
Yeah,
And
exactly. From excitatory to excitatory.
excitatory, yeah, it was
Yeah.
onto a pyramidal, an excitatory synapse onto a pyramidal
Yeah.
neuron, I think.
Yeah.
Um, and and that was all people had, and also it was sort of considered to be the only. I don't know. I'm kind of probably making people look more dumb than they were. It was, I think, the economy of the finding dictated that theory focus on the excitatory synapse, because the experiment could dictate, could only look at the excitatory synapse, because everything else was so sparse that you would have to look for a long time to get reliable data, and it wasn't feasible within the time frame of a single graduate student to actually get results.
Exactly. So it was mainly like experimental constraints in some sense.
Yeah, that's right. I would say so. I would
Good.
say
And
so.
then there was like these different rules that were sort of suggested, right? Like there was like a two
So there was, I mean, there was a
rule.
lot of work that was done. If you Google or if you go onto PubMed, there's 50,000 publications on synaptic plasticity, um, and and a lot of different rules started surfacing. There was LTP rules and LTD rules, and
Mm.
rules that were temporally precise, rules that were
So LTP is
anti-Hebbian,
just long term. What
long-term
long-term
potentiation and
potentiation.
long-term depression. Yeah.
Yeah. Okay. Mm. Yeah.
And in 2000, there was a review published that was called "Taming the Beast," where the
Okay.
beast was synaptic plasticity. I think, truth be told, we probably still haven't tamed it. If anything, the beast has become more multidimensional. But I think there's hope.
Yeah.
We'll see.
Yeah. So okay. So then these rules that came up, there was like, for one thing, they were sort of like phenomenological. They were not sort of based on, at least to begin with, not based on, I mean, the biophysics of the synapses or the biochemistry or what have you, right? They were sort of different kinds of phenomenological rules. Was that mainly?
So, I mean, I like to, people are probably going to sort of hit me in the head, but I like to look at this from the perspective of the Mars levels, like the
Mm-hmm.
computational, the algorithmic, and the implementational level. And I agree with you that those rules are squarely in the realm of algorithmic solutions. They don't really consider any of the implementational parts. They don't consider what we need as biochemical sensors or as biochemical messengers or any of that was sort of kept outside of the parentheses that the field focused on. They simply said, like, we're just going to work with spikes and we're going to keep all the biochemistry outside of those considerations. And
Yeah.
I think to a large extent we still do. I think that's kind of our privilege as theorists, that we can reduce a synapse to just a single number.
Mm. But I guess also, I mean, if you start, I mean, there are models which are sort of more biophysical, but then you're sort of inside this like signaling pathways inside synapses, and it gets really, really complicated models, right? With many players, so it's not so.
It's dirty.
Yeah, it's actually it's a sort of a dirtier kind of. If you want to do biophysically detailed things, it's sort of dirtier than if you just want to do sort of like modeling of membrane potentials and sort of a cable equation kind of things.
Yeah, I
Mm.
think to a large extent there's two problems. Number one is computational efficiency. So it becomes very tricky to model all of this, or it used to be. I think that's maybe not a limitation anymore, but for the first 10 years after CDP came up, it's just, again, I think science doesn't necessarily move towards the question that needs to be answered as much as towards the question that can be answered.
Absolutely.
Um,
Hmm.
and so, even if or when we recognize that there are questions that should be answered, um, it's sometimes quite difficult to then go for that question because it's a hard question, or there's other questions that are more simple to solve. And so it was certainly a much more easy approach to first focus on the algorithmic solutions and then go through the logic and the numerical consequences of implementing those rules in networks. And certainly it's more fun because you don't have to fudge as much, right? As you know, the more parameters you introduce into a model, the more freedom you have, and then at the end of the day you get the answer out that you want to get out, and you can't really be certain that you haven't cooked it by way of confirmational bias.
Oh. So then you ended up with this. I mean, there was like the simplest rules, two-factor rules, which are then, I guess it depends on the pre-spiking, presynaptic spiking, postsynaptic spiking. But then they started also making these three-factor rules, right? You wrote
Yeah,
one.
so I mean, Wolfram Gerstner basically stayed in the field, and I think a lot of the people that still work on plasticity have
Yeah,
gone through Wolfram's lab in one way or the other. So he's really been
I think he's.
very influential.
Yeah.
And he started thinking about two things. He started thinking about, number one, why would only the pre and postsynaptic spike matter in the immediate temporal vicinity of the spike, or of the postsynaptic spike? And he started working on what he called triplet models with Claudio Klopath and Jean-Pascal Pfister and Eleni Vasilaki. I think those are the three people that were most involved in that.
Mm.
don't crucify me if I forget someone, but
No, no crucifixion here.
possibly in reverse order. I don't remember.
Yeah.
And he showed that you get very nice, sort of more stable rules out of
So a
these kind of
triplet
triplet
rule means
constructions.
that, what is that? Does it mean three spikes?
You don't only take the first presynaptic spike that comes along into consideration, but you have a longer memory trace. Basically, you have longer and shorter timescales that are playing a
Okay.
role in the
So
same
maybe like
synapse.
the two spikes before the post batters or something like that.
Exactly. Yeah. So the simulations showed that when you had, I think I might say something wrong again, but I think the simulations showed that if you had a lot of pre-spikes coming together in very rapid succession, that
Yeah.
the model started going a little bit off piste, to say the very least, and that you could remedy that by sort of keeping track of spikes with slow and fast time memory traces at the same time.
Hmm. Hmm. So there were
Uh,
also these. Yeah. Go ahead.
Three factor, you were just about to say, right?
Yeah, three factor, because that's different from a triplet, or it could be
Yeah.
three
As far
others.
as I know, that also came from Wolfram, and that's basically the idea that's relatively well established in that synapses are modulated, in addition to just serving as faithful propagators of information. They are being turned on and off or modulated in their efficacy by additional neuromodulation. So you can regulate
So,
whether a synapse should be. Go ahead.
the membrane potential on the postsynaptic side, that's a factor that's sometimes included, right?
That's right.
Yeah.
I
So that. Hmm.
That was a different. I think that's not the third factor, as far as I know. I
Okay.
think the third. I mean, there's many third factors, right? But
Yeah.
um, the third
Yeah.
factor that I'm talking about would be a neuromodulator that sits in a tripartite synapse and
Hmm.
modulates whether a synapse can be plastic at
Okay.
all.
Hmm.
What you might be talking about is a voltage dependent rule, which is also
Yeah. Exactly.
Gina Trigiano's lab did a lot on that, and Claudia Klopath actually was quite involved with that as well,
Hmm.
with Wolfram.
Cool. Anyway, so this was sort of uh, yeah, this was sort of obviously important, well, still an important activity. But then in 2011, really remember this. It's not that many papers that made sort of like an immediate impact on me. But then you and I think Henning Sprekler, and was like the two lead
Yeah,
authors, and also with Friedman Senke and Claudia Klopat and Wolfram, then in the lab of Wolfram, came
yeah.
up with this really cool idea about what could the role of what the inhibitory plasticity could be, sort of like the plasticity of inhibitory synapses.
Yeah, I mean, I was not a synaptic. I still kind of I don't I find myself feeling like a visitor in the field of synaptic plasticity still. Even though now we've had papers on synaptic plasticity,
You're
quite a
like
few of
you're
them.
like an intellectual imposter.
Yeah, I don't know, but certainly in 2011, I was a visitor,
Yeah.
and actually that the idea for that paper was much older. The idea came around because Claudia and I spent some time together in a summer school in Freiburg, and Claudia was working on these voltage-dependent rules that you just mentioned,
Yeah.
where the postsynaptic side, senses its membrane potential as much as the pre and postsynaptic spiking activity.
Mm.
And Gina Turgiano had shown that that was an actual rule that was implemented at inhibitory synapses, and I had just come out of a year of painfully tuning excitatory and inhibitory synapses. So I published a paper for my PhD that relied very much on excitatory and inhibitory efficacies being roughly the same,
Yeah,
or
because this is sort of like an important idea that obviously came also, well, maybe 10 years early, this idea of a balanced state.
that's
Uh,
right. That was
yeah,
Carmen
exactly.
Briesvik and Reimso and Polinsky originally.
Yeah.
That
Mm.
and that has actually been quite a
Yeah.
powerful
Yeah. That's one of the.
concept.
Yeah. So it was sort of essentially you were, and that seems to be sort of like seen in this at this some of the states in the brain, or maybe like this
I would
asynchronous.
argue everywhere, but if you're a
Yeah.
hammer, the world is nails.
Yeah.
Um,
Anyway, so no.
I certainly think that
Mm.
excitatory and inhibitory. I mean, balance is really not a hard thing to do if your synapses are in the right position, in the right parameter regime, because you have the membrane potential that's buffering the currents, that's buffering the efficacies of the currents by way of the driving force, such that excitation and inhibition will balance each other out quite readily if you're in the right parameter regime. However, getting into that right parameter regime in a spiking network with 10,000 neurons is actually quite painful, and it cost me a good 6 months or 7 months of my life. And so when I met Claudia, she had these plasticity rules, and she was working on excitatory plasticity rules. And I had still PTSD from tuning, and I had read this paper by Gina Trigiano, and so I mentioned it to her, and I said, wouldn't it be cool if you could implement that in inhibitory synapses? And she was done with her project for summer school, and she had a few days extra, and then she implemented it, and it worked right off the bat. So it magically created excitatory inhibitory balance in the neurons that she simulated, and so she's done the very first simulations on all of this work. And then we sort of put this onto the back burner. I was in a postdoc in New York, and Claudia was just about to finish her PhD with Wolfram and had her hands full. And so we didn't really work on this. This was 2007.
Oh,
that's
early.
Yeah, and it's been sitting. It sits on the bench, and I have other things to do, and she has other things to do, and then I moved to, by way of serendipity, I moved to Wolfram's lab in Lausanne in 2010. And Henning Spreckler, who is an old friend of mine from when I was a physicist in Berlin, is about to leave Wolfram's lab. He was a postdoc in Wolfram's lab, and apartments are notoriously hard to come by in Lausanne. So he offered that I move into his apartment for 6 months, and we overlap and live in a shared flat.
Okay.
And then he leaves, and I take over his apartment. And that seemed like a great idea, also because my partner at the time, or still my wife, she was going to stay in New York a little while longer, and so it was the perfect situation. And so Henning and I were in a science camp, so to speak, for 6 months, where we went to work in the morning together, and then went back home and had drinks and talked about science until we went to bed.
I see.
And one of those evenings, I told him this idea about this inhibitory synaptic plasticity that was so magical at creating balance, and he sort of picked it up and looked at me, and I was already 2 drinks in, so I went to bed. And then I got up in the morning, and he stood pretty wild-eyed in my door frame of my room, and basically told me, like, this is working. I did all the math, and this is a cool thing. This is stupidly cool, and I can't believe no one's done this yet. And I was like, okay, whatever. And he said, no, not whatever. You're going to work on this, and you're going to work on it starting today. And if you're not going to work on this, then I'm going to steal it from you. And so I was like, okay, Henning, I guess I'll start working on it. That was the 10th of August or something in 2010. And it must have been a little earlier, I think, because Henning's daughter was born right around that time. So Henning was a first-time father, and was on, like, soon after, went on paternity leave. And then my wife became pregnant around the same time, and started moving to Lausanne, and we were sort of in this strange fight against nature that we wanted. This was a really nice story, and it was unfolding quite beautifully, and Friedemann Senke started getting in on it, and he did all the recurrent spiking simulations. And Claudia was obviously still in the picture, and so we had a very nice thing going, but we had also time pressure on ourselves, and so we started working on it quite hectically. My daughter was born on the 25th of May 2011, and then we sent the paper out on the 13th of July 2011. So it was all very hectic and very intense time. And there's good things from that and bad things from that, I think. You can interrupt me anytime if I'm talking too much.
No, no. This is please go ahead.
The cool thing was that the paper, we sent it to Science, and it went into review, and then it came back from reviews 80 days later. So nothing, and it came back from reviews, and one of the reviewers had simulated everything that we had done. We hadn't supplied any code, but he had reproduced all of our findings, and in fact pointed out there's a mistake here, there's a parameter switch here, but I think it's this and that. But all in all, this idea is very cool. And then the paper was basically accepted, and we had to do all the little things, all the homework plots that we had to do. And then we made a crucial mistake that cost us a good amount of time, which was to not be careful with our citations, because there was a funny thing that happened in the year that we published that paper, or that we worked on that paper, in that originally, when we started working on this, there's a paper in 2006 by not by Julie Haas, by Melanie Wooden, that showed up in Neuron, and that shows, or seemingly shows, the plasticity rule that we were looking for, which is a Hebbian symmetric rule. So pre before post, or post before pre don't really matter.
Was it an experimental paper, or?
It was an experimental paper, yeah. And then it had this, I guess we haven't talked about this rule yet, this magic plasticity rule that Claudia implemented, that Henning then changed. That rule is a Hebbian rule, meaning it's a rule that's symmetric in nature. So pre before post, or post before pre, within 80 milliseconds proximity, temporal proximity from one another, will lead to potentiation. And then anything that is only presynaptic, so any presynaptic spike alone, will lead to depotentiation. And that means basically that you have a system in which a number of presynaptic spikes alone will balance the effect of a pre-post pair, so that when a presynaptic spike arrives from an inhibitory synapse at an excitatory neuron, or any neuron, and there's a postsynaptic spike, then that synapse did a bad job, right? Because the presynaptic spike should have prevented that postsynaptic spike. And so in response to that, you strengthen the synapse, because you say, well, that clearly was a failure. Next time around, please prevent that postsynaptic spike. And so you do that until there's no more postsynaptic spikes, but then the synapse is in a funny situation in that it can no longer distinguish between whether it's completely overshooting and it's way too strong, or it's just strong enough. And so the only way out of this is to create a weakening every time there's a presynapse alone. So if you have presynapse alone, it goes down, it goes down, it goes down until there's a pre-post, and it says, oh, well, that was too far down. Let's strengthen it again. So you have this constant going back up and down. Now that rule, if you plot it in spike in the plasticity window, so on the x-axis you have t pre minus t post, and on the y-axis you have the delta w, so the change in efficacy. That rule looked very much like what Melanie Wooden published in 2006 with Moving Poo. And unfortunately for us, that was not actually true, because what she had decided to do was plot she was recording her synapses as minus 90, and as a result of that, she had two differential. She was showing that there's inhibitory synaptic plasticity that occurs due to two different effects. One was a change in the efficacy of the conductance. So the conductance of the inhibitory synapse itself became bigger. And the other way was that the reversal potential of the synapse locally changed. And so the reversal potential actually went up, so that synapse at minus 90 looked like it was getting, no, the other way around. Hold on, now I'm getting this confused. Doesn't matter. It looked at minus 90 differently from how it would look at minus 70, because the reversal potential of the inhibitory synapse is at minus 80. And so you were looking at the same thing from the other side of the window, basically, that you look at it usually from. And so what we had was not weakening in the temporal periphery, but also strengthening just by a different mechanism,
Okay.
by way of a change in reversal potential, by change of a local chloride concentration.
Yeah. Mm.
And so we misinterpreted that initially, and I gave a talk about this. And some people saw this and then looked into the paper, and we, one of them was in my lab actually, Alan Woodruff, a lab mate of mine at the time already before I moved to, was a lab member in my previous lab and told me about this,
Okay.
um, and and a few other people told us as well, and we were like, oh yeah, that's right. That's actually
Mm.
not the rule we were looking for.
No.
And then we originally had this cited differently, if I remember correctly, but in the process of consolidating all our citations, because if you get a science paper, you originally have 80 citations or whatever is necessary to cite for your work, and then the science
Mm.
editor says, oh, your paper is ready for publication now, please make sure that you only have 30 citations.
Really?
And so you cut out all,
Oh, really?
everything that is not
Yeah.
necessary,
Right.
by any more stringent, at the beginning you're generous, you're like, oh, I should cite this, I should cite this, and then they make you cut 50% of your
Mm.
citations. And in that
Oh,
process, that nuance, that the wooden paper was actually not what we were reproducing, uh, and that it was working regardlessly, that we were making a hypothesis rather than experimental validation, or validation of an experiment, I should say. Um, that nuance got lost in our citation structure. And then someone rightfully spoke up and said, yo, I just saw your paper. This is not right.
so, mm.
And my, we were being pushed very hard by science to get this right. They wanted to, not to get this right, to get this out.
Okay.
They wanted us to publish this before the end of the year, and there was speak of a competing paper that might scoop this, and
Really?
so this was actually the only time in my life that I worked 2 times 120 hours in 1 week in a row, just to get this stupid paper finished. And we didn't really fix it to the satisfaction of the person that had spoken up the first time.
Mm.
And it got published in a way that still wasn't cool, let's say. There was still room for interpretation. And and so a little while later, that person had told another person, and that person had told another person, and then we got an email from someone that said, yeah, we've looked at your paper, and obviously the citation is wrong, and we looked at it and it's all wrong. The logic consequence of your misappropriation of this experimental source of your rule is a cause for concern, and you should retract the paper.
Really?
and and, and at first, obviously, I was like, ah, and then
Uh,
I looked at it, and it was really this way, the way that we had cited this wooden paper
uh,
was just
uh,
stupid, in my opinion.
uh,
But this other side, which was what I would call in hindsight, somewhat unforgivingly, a slightly overzealous graduate student.
mm.
I hope he doesn't hear this now. Um, but I mean,
Yeah.
I don't know. He might share this opinion now. But in any case, he wrote a technical comment that basically he wanted published in Science, and that said,
Mm.
uh, this paper is shit and it should be retracted.
Really?
And and then we tried for a long time. They have to show some communication with us, and then we tried for a long time to find an amicable solution to this, and it really didn't look like it was going to be amicable. And at some point, Wolfram was like, "Okay, let's just play ball." And
Mm.
so we wrote a 15-page response, and basically said this and this and this, and yes, we fucked this up. The
Mm.
citation is stupid. But no, it doesn't actually break the logic consequence of this paper, and the simulations work out quite nicely. And so then they sent this off, and Science sent it to the reviewers, and then it was really to our benefit that one of the reviewers had simulated the paper,
exactly.
basically for us. So he knew all of the
Uh,
model
uh,
inside out.
uh,
He was the one that had done all the homework and told us where all the parameter problems were to begin with.
mm,
And so the predictably the technical comment didn't fly and it was rejected, but it cost us enormous amount of energy.
uh,
So I think the moral of the story is really be careful when people tell you that you're making mistakes.
mm,
Really be careful,
yeah.
because it costs
And
enormous
also
amount of
be
energy to
careful when you
fix that.
sort of pushed under under this pressure to publish quickly for some kind
Yeah,
of when a journal is pushing you, right? Then
yeah, yeah.
that sort
Fair
of is
enough. Yeah,
yeah, that's
and
also
maybe
yeah,
push back, right? Because for the journal, it's also a pain in the butt. They don't want to
yeah,
be
yeah,
going through some sort of correction
yeah.
process. But
So the problem was really not. It was sort of how we had described the link between the model and the experiments that were available at the time. That
that's
was
right.
sort of so
That's
that was
right.
sort of
And you can still, I mean, there is a version on the web that has the
mm,
pre-correction. It was published with the way that we had cited
yeah.
it, and then you can see the difference between the corrected version, and the
Okay.
original version, and then you can make
So
up your own mind whether I
okay.
was.
Anyway, but the model itself, if I don't care about the if we don't care about the experiments,
You
that's
said that.
yeah,
I didn't say that.
life, life gets much nicer. No, anyway. So anyway, but what the model does, for one thing, it's obviously it has consequences for how to think about network dynamics and so on. But the model itself is very easy. It's just it's actually a single neuron, right? That gets synaptic inputs. So
It's
it's
it's stupidly simple. Yeah,
well,
it's
and that of course is makes it easier to understand, and also for this reviewer to reproduce it, right? I mean,
yeah.
if you're
So
a
the paper
reviewer
has
on
four panels, and
uh,
three panels are three figures. Three figures of the four figures are made with a single integrated fire neuron, basically.
that's sort of, I think that just adds to the beauty, I think. But anyway, what could a model model do? It could make it, then the model self tune to get into this balanced state, right?
Yeah,
So you and
basically, it made sure that the postsynaptic firing rate was at
yeah,
whatever we wanted it to be at a set rate. Because
yeah,
remember, I told you that this plasticity rule sought out a balance between number of presynaptic spikes alone and number of
mm,
pre and postsynaptic pairs. And basically, that led to a permissible number of postsynaptic spikes.
mm,
And so the rule basically adjusted the postsynaptic firing rate, and then the process produced this balance of excitation inhibition.
yeah,
So it did exactly what I needed to have 5 years earlier or 3 years earlier when I was tuning
exactly,
for 6 months. It
exactly,
reproduced my original work in 10 minutes, basically.
exactly.
What I had,
Oh well,
you know.
that's so. But then I mean, it makes this sort of like these balanced states, but that's like a background state. But it also could store some kind of memories, right? Or
So what it does, it sort of creates an anti-memory, if you will. That's what the News and Views, I think, called it, or something like
oh,
that. Um, it produced so if you put I mean, we still think that most of our memories are basically situated in our excitatory synaptic connectivity matrix.
mhm,
In that, when you make a memory about something, it's probably the collection of your excitatory synapses somewhere early in hippocampus, then later in cortex, that is the physical representation of that memory. And um, that concept, is not necessarily a stable concept in recurrent networks. That is, if you have groups of neurons that are strongly connected with each other, which we
mhm,
are inclined to call engrams now, those groups of neurons in recurrent network models often become permanently active, so they keep firing all the time, and
mhm,
they have, that's a self strengthening process. So you have a runaway excitatory plasticity that can only be tamed by way of putting a lid on it and saying you cannot get stronger anymore. And then
mhm,
Alfonso Renard actually published some beautiful work in how you could, maneuver those engrams and turn them on sequentially and such,
okay,
by giving them excitatory and inhibitory inputs. But what the inhibitory synaptic plasticity rule contributes is essentially that it turns off those ever active excitatory engrams by producing a counter memory, right?
Yeah, I see. Okay, yeah. So, but it also points to some sense like a division of labor between excitatory synapses and inhibitory synapses. That the
For sure.
excitatory synapses deals with the like information content, memory content, and the inhibitory plasticity sort of is like the janitor that sort of helps the that it
Yeah,
doesn't run
if you want
away.
to
That
call it the janitor or the mastermind, is really dependent on who your best friends are, right? I
yeah,
think what I've started I mean, again, hammer nails. If you think a lot about inhibitory neurons, you anthropomorphize them to not be the janitor, but rather the horseback rider, or like you start thinking about this vast pool of excitatory neurons as the cows on the ranch, and then the
okay.
inhibitory neurons flying
Ah,
through there like the cowboys or cowgirls taming those excitatory neurons.
exactly.
Um,
Yeah. So anyway, so this, I mean, the excitatory. I guess in this paper you had this excitatory rules that was sort of from the back in the days. This was before 2011, and then you came up with then this inhibitor plasticity rules that made this balanced state, for example, had this
we
problem.
didn't actually have excitatory synaptic plasticity in our paper, and it was criticized, and it was also criticized later in grant proposals.
Oh,
That obviously we were arrogant to exclude excitatory plasticity, and my response was always, for 50 years we've ignored inhibitory plasticity. Isn't it time we start ignoring excitatory plasticity? But um, um,
yeah,
but
exactly.
in reality, it was just too
So
complicated, right? Again, the economy of science dictates that you take small
okay,
steps. So we just focused on
but
inhibitory
later
synaptic
this has
plasticity.
sort of been maybe then
So
combined.
Friedemann Senke, who was one of the authors in the science paper, we did all the recurrent
Also, a
network modeling.
earlier podcast guest in theoretical
Yeah, all right. So he ran with that idea, and he implemented recurrent networks that had both excitatory and inhibitory plasticity. And he showed that he had a slightly different inhibitory rule, but he showed very nicely that you could use inhibitory plasticity to keep excitatory plasticity in check, actually.
understanding. So has there been any sort of like post hoc sort of confirmation or testing of this inhibitory plasticity rule?
So there's two papers. One is from 2015 by Rob Fremke and James Damore, who very kindly spent their time, running
Mm,
plasticity experiment on inhibitory synapses, which still is not
yes.
easy because there's
No.
so few of them.
Yeah.
And they saw a rule that is somewhat reminiscent of the rule that we postulated. So with temporally proximal pre-post pairs, um, um, potentiating and temporally distal pairs, or if you will, just presynaptic spikes alone, even though that's not what he's shown,
Mm,
um, um, because he has a pre-post pairing, right?
mm,
Weakening.
mm.
So we saw some experimental confirmation in 2000, I think 15 or 16, and then there was another paper that is still under review and hopefully out soon, by Anne-Marie Oswald and Brent Doiron as the last authors,
Mm.
who've done experimental work on two different types of synaptic plasticity.
They're in Pittsburgh, right? Or they used to
That's
be in
right. They
Pittsburgh.
used to be in Pittsburgh. They're now in Chicago.
Okay. Mm. Yeah,
They recently
exactly.
moved, I think, during COVID, also 3-4 years ago.
Mm. Yeah. Cool. So then, I mean, okay. So then, after I was at like 2011, and then your paper, and then also with Friedemann Senke, who showed that you can actually combine these excitatory synaptic rules with some kind of variation of your inhibitory synaptic rule,
That's right.
and yeah, but they were sort of then independent rules. But now you in this 2024 paper, I guess with Everton Agnes.
Yeah.
Yeah,
Everton
then
Agnes, who also was a co-author on Friedemann's paper, right? So
oh, okay.
Everton Agnes and it was the second author on the Zenke 2015 paper, because
I
Everton
see.
was a visiting student in Wolfram's lab,
Oh,
published something with Friedemann, then went back to Brazil, finished his PhD, and then
okay.
worked as a postdoc in my lab. Um,
Yeah.
and kept working on this. And his idea was basically that the interaction of excitation and inhibition doesn't have to be by way of network effects, like Friedemann showed, and Everton helped show, but also
Mm.
that this could happen immediately at the same synapse or at, ambient synapses. So he started looking into whether we could explain the results that we see. There's sometimes, conflicting results of different excitatory synaptic plasticity findings, whether we could explain these if we considered inhibitory synaptic plasticity sort of as a guards, as a groundskeeper for excitatory plasticity and vice versa. And so he postulated two rules: one for excitatory synapses that had excitatory synapses being gated, by inhibitory currents, and then vice versa, inhibitory synapses that were being gated by
And
excitatory currents.
and here we think about sort of like actually two synapses, an excitatory and inhibitory synapses being co-located on a dendritic branch or in a
They
spine.
are actually.
Yeah.
So I mean, excitatory synapses on pyramidal neurons are mostly on spines, right? On at least on corticopyramidal neurons.
Yeah.
But Corette Vierenga from the Netherlands, she showed in quite beautiful work that excitatory synapses are actually often accompanied by inhibitory synapses that are sitting in the close
Okay.
vicinity on the
Also
dendritic
on
shaft.
cortical neurons. Yeah.
Yeah, yeah, yeah.
Yeah. Exactly. So then you have this sort of like this exactly this biological setup for this kind of codependency where they actually
That's right.
modulate each other. Yeah.
That's right. Yeah.
Yeah. So that's of course then it's then you have two rules. Well, in you start with one rule for the excitatory, and one for excitatory synapses, one for the inhibitory, and then you connect them. So this becomes this quite like a more complicated model.
It's more complicated in the implementation,
Hmm.
but it actually turned out to be much less complicated for the tuning. So excitatory synaptic plasticity alone turns out to be quite unstable and finicky in recurrent networks. That's why
Okay.
Friedemann built his inhibitory
Yeah.
synaptic plasticity, and then when you build excitatory and inhibitory synapses that are plastic, you have to often think about how you stabilize them separately, because
Okay.
excitatory
Yeah.
synapses will have a fixed point that they run towards, either as a weight fixed point or a rate fixed point. And similarly, inhibitory synapses, at least under the plasticity rules that we've implemented, have have a fixed point in firing rates. And so if you assume that they always know of each other and they know exactly where they want to be and they coordinate by
Mm.
talking to each other in whispering voices, then that's no problem. But as soon as you assume some sort of biological noise, you can show quite nicely that excitatory and inhibitory plasticity, at least in the combinations that we've implemented them in, they don't play very nicely with each other. They
Okay.
start competing. They start breaking
Mm.
each other's
I
style,
see. So it's
um,
actually
or cramping it.
easier if they're married together in some sense.
Exactly. And so the codependent plasticity rule, that's why we called it codependence, right? And in fact, the editors were like, oh, codependency is a term from psychology and it's not a good term. And we're like, well, for
Yeah,
synapses,
exactly.
maybe it is. So because
Exactly.
then they have a chance to communicate with each other,
Yeah.
simply because they are, they explicitly rely on each other rather than by way of sort of independent measures.
By the way, when exploring these models, but also the 2011 model, you both used like spiking neuron simulations and firing rate models typically, or
Um,
how was that?
so I mostly use spiking models, and I've used rate models only very rarely in my own
Mhm.
coding, in my own
Yeah.
work. Um, Strangely, I think I love spiking network models. They're fun
Mm.
and they're finicky, and they reward you quite nicely for persistence.
Okay.
Whereas rate models are much more agreeable with you, right? They do what you want quite quickly, but I never had a good intuition for what to do next, because it seems like the solution came too quick, and I never had time to think about, when you when you're tuning spiking networks, you have a lot of time to think
Okay.
because because they're so freaking
This
hard
is
to tune.
this is sort
Whereas
of
with weight networks, the solution
this is the
arises,
funniest
and then you're like,
argument for using spiking neurons I've ever heard.
I'm a little stupid, so I
Uh,
use hard
well,
things
if you
to.
want more time to think, you could go to multi-compartmental models.
That's right. Yeah, but multi-compartment models again have this fudge factor in there, right?
Yeah,
There's too
that's
many
the-
solutions
yeah.
out there,
No, that's
and
mm. Yeah,
so I was yeah. The sweet spot for me was single compartment spiking models,
yeah,
and then
excellent. Mm,
Henning is obviously mathematically much more crafty and able than me, and he did the parallel work analytically
mm.
and implemented some of it in rate models for the 2011 paper.
Okay.
And in the
Yeah.
24 paper that with Everton equally is mathematically um, quite quite obsessive,
Mm,
and so
yeah.
in fact sometimes to a fault,
Yeah.
because spiking models show that the mean field models aren't always reliable
Mm,
in
sure.
predicting the dynamics of spiking networks.
Mm,
So it was always nice to have someone else do the math that I really didn't trust myself doing, and then comparing the implications of the mathematical models
yeah.
with the numerical
Mm.
simulations.
So what did you so now this codependency model is, well, you learn that it's actually sometimes easier to tune the model, maybe also meaning that it sort of is also easier for the real system to actually sort of to come up with a good solution, a good functional solution for the in the cell. So what were sort of the main insights or sort of that you got from the codependency paper?
I mean, I think the main point of that paper really is that um, your if you don't have a way of making plasticity rules talk to each other
Mhm.
um, nicely, they won't play nicely with each other.
Yeah. Mm.
That's sort of the take home message, which
So, mm,
which was known before, but we just made that explicit. I mean, this is not coincident that Friedemann's paper in 2015 had the word orchestrated in the title.
I see
Orchestrated just means we spend a lot of time tuning it.
that you,
Um,
you exactly wasted a few graduate students sort of to
yeah, well, just Friedemann,
that's the way. Yeah.
yeah,
Okay.
yeah, yeah.
Wasted Friedman's resources. Yeah. So essentially, if you want to, what you're saying, if you want to have both an inhibitory and excitatory synaptic plasticity in a network model, say, then you might as well add this codependency to make life easier for you.
I think that's fair to say, even
Yeah.
though in our own work that we in our defense started before Everton's paper really got going. We went
Mm,
back to models that are not codependent as a first step. So we went slightly different route, but we're catching up with the 2024 paper in a way in my own lab because we started thinking about um, doing the search for plasticity rules, this iterative process of finding the right parameters for, I mean, we sort of changed tuning problem. In 2007, I had a tuning problem of tuning excitatory and inhibitory weights.
yeah.
And then I fixed that by creating a plasticity rule together with Henning and Claudia and Friedemann that took that tuning problem away. But then very quickly, you go and realize that actually the excitatory and inhibitory rules need to be tuned, right? You need to
That's right. Yeah.
finally orchestrate them. So you just moved your
Exactly.
tuning problem.
Mm,
And so one answer to that tuning problem is the codependence, and the other answer is let someone else do it. And by someone else, we figured we let computers do it. That was at the time when 2018, 2019, when the ML assisted automated discovery avenue was quite new in its at least in its
yeah.
um, in its might, in its computational might. And
Mm,
so we wrote a grant and said, we're going to do all of this, with computers, and just let this computer figure out what the rules are. And
mm,
why don't we do it not just for excitatory, but we do it for all four of the rules. So we'll build spiking networks with two synapse types and two neuron types and four synapse types, and we'll let the computer run and
mm,
see which plasticity rules are the right ones.
mm,
So that was the avenue that we've been pursuing, rather than focusing more on codependency. Of course, the next question is, why didn't you add codependency as a factor
mm,
into all your rules, which we should have, but
mm,
that was sort of the boundary of what we could do. We had four rules.
mm.
Each of them had six parameters, and that was 24 parameters already. And so adding more complicated rules into the game was something that at the time
Mm.
wasn't
Then,
possible.
yeah, so there was a paper that came out this year, actually, PLOS Computational Biology, Basile Confavreux
That's
as
right.
the first author, and then, well, Everton and the usual suspects, Friedman, Henning, and you, I guess.
Yeah,
Yeah.
yeah. So this paper is kind of what we thought was going to be Basile Confavreux's PhD work, because he tried for a long time to meta learn plasticity rules. And so what I just described, that you
Meta
do
learn
this.
means that you want to sort of look on find these learning rules by
Yeah, it's
trying
called meta
things.
learn because learning is usually attributed to with
Yes.
plasticity, and then the meta learning is learning of the learning rule, right?
Yeah.
So it's
Mhm. Yeah.
learning of the behind the scenes sprockets. Um, and he tried this idea that we had gotten this grant for 4 years, 5 years,
Mm,
and what you see in the PLOS paper is the result of that search,
mm,
where we use genetic search algorithms basically to find the right 24-dimensional parameter combination
mm,
to learn stable networks,
mm,
networks that simply didn't blow up. That's not the end of the story, thank goodness, because um, we had a very serendipitous interaction with a grad student and a postdoc at the Macke lab in Tübingen.
mm,
Um, this graduate student, Purnima Ramesh, she contacted us early on after we had a couple of papers on this sort of meta-learning approach that Basile
mm,
was working on in 2020, and NIPS was one, and then this 24 paper in PLOS Comp Bio, and there was a few other iterations at conferences.
mm,
And she contacted us and wrote us a very polite email and basically told us that was a stupid idea what we were doing, and it wasn't going to work.
exactly.
Very nicely, she's a very polite woman, but it was very clear that she didn't think so highly of our approach, because she could
No
see it
comment.
was going to fail.
Mm,
And in fact, Basile had come to the same conclusion already, because he
mm,
had seen it fail. I mean, this was 2 years ago, 3 years ago, and we were like, well, thanks, Purnima. And then she was like, well, actually, I think I can help you. And what she thought at first was going to be the holy grail
mm,
was actually a different model from what we ended up really using. She had some sort of adversarial learning scheme in mind, but in the process of trying that out, um, it surfaced that Purnima and Basile really had a very good channel of communication. So they both were, Purnima was doing the computer science and the implementational side, and she had all the computational tricks up her sleeve. And Basile knew all the synaptic plasticity literature, and they started sitting next to each other in Tübingen and in Austria, and just interacted and became, quite indistinguishable from one another, and that you could always ask one person where the other one was
mm,
with their code, and so they came up with a very beautiful implementation of work that Pedro Consarge, who's now a PI in Belgium, but who at the time was a postdoc with Jakob, and who had already collaborated with us
mm.
on this technique that he had developed, which is called simulation-based inference.
Yeah.
And he and Purnima and Basile basically developed that technique further to really work beautifully with exactly what we had in mind, finding parameter combinations that would grant us plasticity rules that can learn things, that can at least create stable networks. The trouble was that instead of finding a single solution, Purnima and Basil basically found thousands of them, literally thousands. So they found a manifold of solutions, of plasticity rules
So why is that
that
the
were
problem?
all.
Isn't that good news?
Is that good? I don't know. I mean, now we
I'm
have the
just
opposite
thinking about.
problem, right?
Yeah, well, I mean, I think it's sort of because we're so. I think this sort of like this tradition we bring with us from like physics, where you typically had models which was sort of whatever simple, low dimensional, and with so few parameters that you could sort of fit them to a unique solution and unique parameter set was sort of much better than the others. But
Yeah.
I think in biology, right, the many solutions means robustness. So
Yeah,
there's
I mean,
something
Eve
to that.
Marder's work, right, on
Yeah,
degeneracy is
yeah.
exactly in that avenue. So we're not complaining that we find so many. There's
Oh,
definitely many questions, but
but
of course, when you have
if you write the
2,500
paper,
different.
yeah.
Say again.
No, man, if you want to write the paper, and it's like a cleaner story if you have one solution, right?
Exactly. I mean,
Yeah,
before, you publish a paper with one rule, or
yeah,
Friedemann had two rules, or
yeah,
Everton had two rules, and now we were hoping that we'd have four rules, but we actually have 10,000 rules, right? We
yeah.
have
Oh, well,
10,000
yeah,
individual rules that are working in pairs or in quadruplets instead of
mm,
four. And
yeah,
so how do you now that's, if you have a one page per quadruplet,
mm,
that's 2,500 pages of supplementary materials.
yeah.
um, I
But
don't.
for,
We're
but don't
working
you
on
really
it.
think that, of course, you're making this say if you have your loss function and you have like two and a half thousand solutions, and probably if you add something more to the loss function, you maybe go down from maybe the rules becomes fewer, right? Because it's
Yeah,
always
we haven't quite gone there yet. So, I
uh,
mean, we haven't certainly. I mean, what you're saying is certainly true. So, what we've started doing is we find 2,500 rules that can solve the problem of stability. So these quadruplets all solve the network stability issue, that these networks don't blow up and they behave nicely. And then Basile started giving them tasks. So a familiarity detection task. You stimulate one set of neurons, you stimulate another set of neurons. You ask after 2 hours whether the network responds differently to a familiar set of stimuli than to a totally novel. Or you can ask, what happens if I stimulate sequences? So, I play this and then that and then that. Can you resurface that sequence in any direction? Can you go forward, backward, whatever have you?
mm,
And so you're exactly right, right? Like if you want sequences, your solution space collapses to like 5 rules.
yeah, exactly.
If you want novelty detection, that's actually pretty easy. Many rules can do novelty detection. If you
Uh,
want omission detection, like you play a sequence, you leave one out. Does the network respond differently if you leave one stimulus out? We can do that now with spiking networks, which is kind of
yeah,
amazing.
yeah.
It's totally like outrageously unthinkable that we would be able to do that in 2011, and now,
Yeah. So there
Basil.
are like exactly. So now for this, because you do it with some kind of obviously some recurrent networks of spiking neurons, and
Yeah.
how big are them typically? How big do they need
They're
in
either
order?
5,000 or 10,000 neurons. So I mean, the Zenki model is essentially the published precedent, the 2015 model that we are using, because the dynamics are known and we have good knowledge of what happens in that model. And we're running it actually on Friedemann's software. So he developed a compiler, a software implementation of spiking networks that's called Orin. That's freely downloadable and is amazing. It's still faster than pretty much anything out there. The Nest people are probably going to write me an email and say that's not true, but in my experience, the Orin is substantially faster than anything else that we've been using, and much less clunky. It's
Then you
written in C++.
yeah. So then you probably need to. I mean, then you need supercomputers maybe to explore these rules, and because you need to run them many times, and
So we used up a lot of processor time. Basil certainly burned through a lot of processor hours. I probably spent about half as much. I spent, if you count the money you spend, I spend uh, a fair amount of on Basil's salary, but I spent about 50%
Okay.
in addition to that. 50% of his salary I spent on compute for the last 2 years.
All
So,
right. Hmm.
so
Did
it
you
was.
do it in some kind of cloud solution, or do you have a local or some kind of super?
We did it at first. We did it in Tübingen. Then we looked into Amazon, but Amazon is incredibly expensive.
Yeah.
And then we got a really good deal at our institute, where I'm at a relatively small institute now. At least we have 80 PIs,
Yeah.
and not all of them obviously use compute, and many of them are now interested in GPU computing and
Hmm.
leaving the CPUs for us to use.
Exactly. Yeah.
So
Oh.
yeah, so it was actually quite a good deal. It wasn't expensive, and also it wasn't a lot of times. Actually, people um, note that burning that many processor hours is also an environmentally
Hmm.
dubious affair. But one of my grad students actually calculated that we used half a transatlantic flight for all of our compute so far, because we
Okay.
have carbon offset
Okay.
and green electricity at
Exactly.
the.
So exactly. So compared to sort of like the maybe like typical use of these large language models, it's still not too bad.
Oh yeah, for sure. And I mean, it's also definitely morally probably more defendable to do this than to have it write your essay for
To cheat
or
on exams. Burn. Burn. Yeah, exactly. No,
yeah.
that's true. But one thing you found is sort of, I mean, you essentially have, I mean, we have excitatory and inhibitory. You have essentially four connections and four synaptic rules. But you can sort of fit to get the sort of this basic, well, these basic firing rates, whatever this background states,
Yeah,
just by varying one of them. So it was sort of quite. So it was like one thing. It was many rules, but you could also sort of have you find them just by freezing three of them and vary the fourth
that's
one.
right. The knockout experiments that we did. So, I mean, that's sort of exploring this manifold, right?
Yeah.
We wanted to know really how stable the rules were, and whether you needed all of them once you found a solution. And it turned out that excitatory synapses actually could work alone in isolation quite nicely for quite a few of these rules. Not the classical SCDP rules as expected; those are unstable.
Mm.
And then we did another set of knockout experiments where we where we took away one out of the four rules and looked at whether the three other rules were able to supply stability to a network. And it turned out that many of the rules actually couldn't. So when you take one rule away, the system collapses, especially if you take inhibitory to excitatory synapses away, or synaptic plasticity,
Mm.
or vice versa, excitatory to inhibitory synaptic plasticity, the system becomes quite labile.
Mm. Exactly.
Sorry.
Yeah. Exactly. So cool. But anyway, so certainly a take-home message is sort of this using machine learning to explore rules, right? That's instead
I
of
think that's the future, probably. I think the times of hand tuning rules are
uh,
essentially over.
yeah,
Um,
yeah,
there's still, there's now going to be, like, when we use inhibitory synaptic plasticity to get rid of tuning
yeah,
strengths, we started tuning the plasticity rules. Now we've taken away the need for tuning plasticity rules, but
yeah,
now we're going to start tuning the search algorithms that tune the rules, right? So the tuning itself is probably not going to go away, because somehow we need an occupation as a human.
yeah.
And there's a question of how you interpret the rules that you get when you include more complex parameters, like
Mm.
codependency, for example, or voltage, or
Yeah.
second messengers, or
Mm.
neuromodulation. There's a lot work left that's still to
Yeah.
do. There's no
Maybe
way
there's
that automated
also
discovery is going to
I don't
save us,
know.
but
I don't know. I mean, if you sort of look at the biophysical level, at these synaptic learning rules, and I mean, there's some kind of transcription of right proteins that makes these channels. So it could be that it's. I mean, I was also fascinated by this work also in with Tim O'Leary, I guess
Yeah,
in where he sort of showed that you could actually, instead of tuning channel conductances in multi-compartment model, or sort of like the Hodgkin-Huxley style models, you could sort of have some learning rules for this transcription or some, and then you ended up with much fewer rules. So in some
yeah,
sense, you change the tuning to a deeper level. So maybe it's something like at some point
yeah.
these your rules would also meet the biochemistry or biophysics, right?
That's right. And I think, I mean, one thing that our results really point at is that we're missing some of the underlying control mechanisms, right? Like we're showing you 2,500 different instantiations of
Mm.
plasticity rules that all work, but how do they get? We know that neuromodulators change plasticity rules, right? We know that probably there is a group of family, like a family of rules that are all sort of flexibly or interchangeable for the conditions that the network finds itself in. But our models do not incorporate the real underlying mechanistic,
Mm.
causally important variables
Yeah.
uh, that make that all happen. You know, and in parallel, there is a discussion now in neuroscience about whether spike timing dependent plasticity is actually the right way to go about learning, whether there isn't a second, more slow timescale plasticity that's called behavioral timescale plasticity, that is essentially able to capture slower learning processes. So, you know, we're not
Do
even
you think it's
incorporating
some
any of
kind of
these.
like some kind of molecular memory thing, or is
No,
that
no, simply, I mean, they're also spec time independent plasticity rules. They're just slower in their time
okay?
constants.
Exactly, slower. Mm. Yeah, cool. Okay, so finally, I thought that we should talk a little bit about another sort of activity that is sort of made
Hmm.
possible by modern day computers. So you wrote this nice comment, I think, which that came out in in Nature Reviews Neuroscience together with
Oh, you
Panos
mean, yeah,
Possibles. Yeah,
yeah.
yeah. So talking science, comma online. Yeah. So what was that all about?
Well, I mean, yeah, I find people like you and me that were alive before the pandemic still actually all have a memory of this of
Hmm.
this platform, Worldwide Neuro, that Panos built
Hmm.
in my lab. It's, you know, now that the pandemic is gone, unfortunately, maybe fortunately, but also sadly in some way, we've all fallen back to our old ways of burning a lot of fossil fuels to go to conferences and see each other and speak at seminars and so on, fly
Yeah.
to conference, fly to interviews. I just heard that someone had to fly or has to fly tomorrow from Australia to Europe to participate in a group interview that's going to last an hour,
Oh, really?
and the funding
Oh,
organization was unwilling to facilitate a online interview situation,
oh,
and so that's, you know, that's a lot of CO2 for 2 hours of interviewing,
yeah.
um, and a lot of money too,
Yeah.
um, and and
Really tiresome.
yeah.
That's yeah.
Anyway, so Worldwide Neuro is a child of the pandemic when we all had to stay at home and couldn't fly and couldn't travel and couldn't see each other, um, and we, you know, Panos was already playing around with code that would make it easier to schedule um, in-person seminars, um, and so we made it possible for other people to access that platform so that they could very easily schedule scientific talks and advertise them on a platform. And we had a bot that tweeted them out. This were the glory days of Twitter. In the heyday of the pandemic, we had 15,000 visitors a day that came back again and again and looked for conferences and looked for scientific content to communicate and talk about, and that was quite cool. I mean, it really made science a much more worldwide affair for a little moment. It gave access to scientific talks to people that would have otherwise not had access,
Mm.
um, and it made it possible to also stay at home and hang out with your kids instead of having to sit in a plane for those 8 hours that it takes you to travel to give a talk or attend a conference or whatnot. And so that's what that article was about. It was calculating the average CO2 footprint of an average conference goer, which is 13 times their daily average consumption. So if you go to a conference, you use 13 times as much CO2 as you do when you don't go to a conference. And then we compared that to the amount of CO2 that it takes you to distribute a talk on Worldwide Neuro, which is obviously no comparison at all, because it's a fraction of that.
But Worldwide Neuro is still up and running. I was up there checking yesterday, and there's plenty of
Of course.
folks
I mean,
from
in fact, we've talked about this. We've been meaning to put podcasts, for example, on
yeah,
Worldwide Neuro, and we're playing with a couple of other formats that I think are still valuable and that are coming, and maybe now in these sad times um, of let's say scientiphobic governance from some countries
yeah,
might come back, in that we're offering a couple of different things. We're offering still, of course, conference hosting. We provide a platform that you can use to crowdcast, to broadcast scientific talks. We're also capturing a part of conferences, of in-person conferences, by way of providing virtual posters. So you can
okay,
upload your poster and publish it with a DOI, so that you can actually have a much more quick way of communicating your science and not being worried about being scooped. That's quite neat. Then we want to open this up to platforms, to podcasts, so that you can advertise podcasts and have all your neuro podcasts or all your science podcasts in one place. And then the last thing that we were hoping to work on, but that unfortunately we don't have enough energy for, because we're a 2-people operation still, and it turns out that energy is the minimizing factor for everything. The last thing we wanted to do was what's called Worldwide Classroom. So sort of a iteration of these mass online courses that Adrian Fairhall has taught so successfully. But giving people the opportunity to just teach one course. What is your best course, Gaute? Can you teach electric brain signals? Here's your book.
absolutely. That would be our specialty, I think.
You give your favorite lecture, or 2 or 3, and then you post that one lecture on Worldwide Classroom, and people can put together their own syllabus, right? If you want the theoretical neuroscience, you start with Gaute, then maybe you go to, I don't know, Jutta Pujarazi talking about multi-compartment models. So we would go to every scientist who wants to teach in the world and say, hey, why don't you give us your best lecture? And then you'd have some sort of much less energy version of these mass access platforms.
Okay.
But we haven't quite got there. I mean, if
How can
anyone
we?
hears this and wants to join,
No, I mean, but there are, at least looking at the statistics from previous podcasts, I think there are more than 1,000 people and listening to each of the episodes, and
yeah,
they also there for the long hauls. So so you reach quite a lot of the community here. So what would be so what could we as a community do, sort of like to support this or to pool
Worldwide Neuro
yeah,
the easiest thing for anyone in the community to do is organize online conferences. So if you feel like you don't have speakers at your university, invite them to give online talks and publish them on Worldwide Neuro, make them accessible to other people, have them recorded so that then they can be archived, and publish them with a DOI if you want. So we offer DOIs for talks now as well. That's the easiest thing to do. The next easiest thing to do is probably send us 10,000 emails asking us for making podcasts available on Worldwide Neuro, because that's kind of already there. We
especially
have a
this
beta
one.
version.
Where,
Yeah,
where is
exactly.
the
No, I
Red
mean,
Gorillas podcast?
a lot of this is sort of need based, and if
Yeah.
Panos doesn't believe that anyone's going to actually consume it, he's not going to do it. And we have now actually 2 people, Panos and Hajer Ali, who's an intern in my lab. She's also working on Worldwide Neuro a little bit. But if they don't feel like it's worth their
That's
time,
true.
then they're not going to do it. And so if
Yeah.
you send us an email and say, oh, I really, really, really want the podcast section on Worldwide
Yeah.
Neuro, then it's
Okay.
gonna happen,
So this
or
is
a worldwide classroom or something like that.
a call to all my listeners to write Tim an,
Yeah,
an email.
worldwideneuro at
Yeah. Oh,
gmail.com.
yeah.
Please send
But
us an
like
email.
several emails a day. Yeah,
Which would be, this is how I met you,
exactly.
Gaute, that
That's
I
sort
had
of
you spammed by SFN. So
yeah,
this is
that's
the
true.
revenge.
Back in the days. Yeah, this is a cool story. Maybe I should tell the story. I think it was a fun story. Tim and I met at the second to last day at the SFN conference, and I was leaving. There was like some a few beers involved in the evening, and I was leaving the day after, so I wasn't going to attend the conference. And then we had the brilliant idea of switching name tags. So you
So we wouldn't
walked up.
forget each other's names.
Oh, exactly. And then on the last day of the conference, Tim went around as Gaute Animal, and then whatever scanned his card at all these experimental vendors. So
It was great.
I still get catalogs. I really feel bad because I get these really nice and glossy catalogs about experimental equipment that is sort of
I
not
got all
so
the
much.
toys and you got
Yeah,
all the catalogs. It only dawned on me afterwards. I mean, you know this, but
yeah.
maybe the people listening don't know this. It dawned on me afterwards that I had done something terrible, and
Well, no, but I
I
mean,
was going to spam someone.
every time I get the glossy catalog, I think about Tim. So about the
Yeah,
fun time. He's sort of a way to get remembered. So
yeah,
yeah, right. So that's good. But because I mean, I was involved in the like on the board of organization of, I mean, organization of computational neuroscience, making this computational neuroscience meeting. And I remember
hmm
in 2020 the meeting was going to be actually in Australia at the time, but of course it was sort of this was going to be in whatever July, so
yeah,
this had to be canceled due to the pandemic. So then we made it online, and it was interesting because it's sort of one thing is that they sort of got much more attendees online, maybe like a factor four, like multiplier compared to the number of people who had been able to go to like Australia, which is a
yeah,
beautiful place. It's expensive and far away for many people. So, and the other thing was not only more people, but we also had people from because I've been to the teaching at the Latin American school in Sao Paulo.
yeah,
I'm going again, and of course that's like a half a billion people in Latin America, and they have like strong research communities, and they typically don't go to meetings in like North America or Europe, right? Because for
yeah,
obvious reasons, right? And suddenly you can make a true global community, as I said. So this is
yeah,
certainly and that was one of the motivation for me for starting this podcast, also to really to have well contribute to a true
yeah,
global community. So this is
yeah.
just something we have to learn how to do.
Again,
Yeah.
yeah, it's hard to do it. I mean,
Yeah.
you can do the science part, and you can sit together like we are now, looking at each other through a camera. It's still not quite as good as the real thing. And we started thinking about that at Worldwide Neuro too, and we actually with Elif Mueller at the University of Montreal, we started writing a grant for three-dimensional classroom experiences and using
Uh,
augmented reality
uh,
to see if we could overcome this. But again, energy is unfortunately finite,
yeah.
and still,
So,
then it
yeah.
just might
But I
might
guess
not
one
be.
ambition is sort of to I mean, instead of having some kind of mixture of meetings, right? That's sort of
Yeah,
that you have some which are physical, and maybe the ones that are involves most traveling becomes more like virtual. I don't know. But we just have to. The perfect should not be the enemy of the good, right? We have to be pragmatic,
yeah,
because
yeah,
it's
yeah, for sure.
especially when you are. Because I remember when I started doing computational neuroscience, the first I was at this methods in computational neuroscience meeting in Woodsole, and that's was a wonderful experience, like 4 weeks together. So
Yeah,
this still forms a big part of my network, right? So
yeah, for sure.
it's really especially when you are a beginning scientist,
The summer
then it's
schools are hugely important. I agree.
so that's true.
So I was just going to say, Dan Goodman and Friedemann Senke again. They're organizing a yearly conference that's called SNUFA. That's online and purely online,
Mm.
and still
Huh?
it still lives on quite successfully, even 4 years after the pandemic.
Yeah, and you were also involved in this neuro match, right?
I
Neuro,
was very tangentially,
yeah, it's
sort of
still
by a
also running.
nature of,
Yeah.
yeah, Neuromatch is still running. I've lost a little bit. I have no insight in how they're doing,
Mm,
because it was, again, I couldn't do Worldwide Neuro and Neuromatch at the same time. I really loved the idea. I liked the people there. There was a lot of energy. They sort of took a very different approach from us at Worldwide Neuro in that they involved a huge number of people to run the infrastructure and to
uh,
provide the content,
yeah.
in a very involved way, whereas we provided the infrastructure sort of in a
Mm,
asynchronous way. Worldwide Neural said, here's
yeah.
the infrastructure, now play,
Yeah.
and Neuromatch was like, build the infrastructure with us, because they had
I
a,
remember that.
for them it wasn't as much of a tech stack, I guess. Initially it was just breakout rooms on Zoom,
Yeah.
and so they had a
Yeah.
much bigger teaching community,
Mm,
and I thought that was really, really cool.
yeah.
That's something I wish we had had with Worldwide Neuro, but
Mm,
I have no experience in guiding like a multi-programmer team, and I was already happy that that
yeah.
Panos
No,
was doing it.
that's no, that's non-trivial.
Yeah.
I would think. I have no idea. Yeah.
And it's not my,
So
I'm a scientist at the end,
yeah,
right? This
yeah,
is
that's true.
service that we did, but
That's uh, that's also the point, right? That sort of is you have to. I mean, if everybody can sort of, well, if many, not everyone necessarily, but many can contribute a few percent of the time to do outreach, like then and not give up the science, because then that's yeah,
yeah.
that's
I
nobody.
think
Well,
Thomas Ryan from the University of Dublin, from Trinity College,
yeah,
said that if everyone contributed 2% of their time, we'd
mm,
be set.
yeah, I think that's true, and of course it's I think it's a lot of fun also. I think it really, if you're a little bit sort of not completely introvert. I think sort of like having because research is often a sort of a when you were sitting there tuning for half a year, it was sort of a
Yeah,
largely a lonely thing.
solitary,
So I think
solitary
to have
activity.
sort of, if you're sort of to have uh, some
Yeah,
kind of yeah, a way to sort of also have a more social thing, and which popularization often or communication often
yeah,
is.
yeah, for sure. And it gives you, I mean, especially for theorists, our work has a very slow time scale of being useful to anyone at all. So these kind of
Mm.
surface exercises, at least, give you
Huh.
the appearance of usefulness for the community.
Cool. Okay, so this is sort of what I had on my agenda to talk with you about. I don't know, is there anything we have
Um,
forgotten?
I used to be organizing a summer school in South Africa, actually, the Imbizo, and you can find information about it on a website, imbizo.africa,
Mm,
or you can just Google Imbizo Africa. And the applications are open right now. So
excellent.
2027, the summer, is in January 2026. Sorry,
Okay.
January
Yes.
2026.
Is that
We'll be
mainly
holding.
for African students or?
It's 20 African. We have spots for 20 African students and 10 intercontinentals. So
Mm, okay.
we're running a pretty diversified operation. Or we, I don't run it anymore. I'm in the
Yeah.
background now. But
Mm,
it's a summer school to network, like you said.
yeah.
You grow your network from these summer schools, and so we didn't want networks that were
Uh,
sorted by continent. We wanted
yeah.
networks that were intercontinental, and so there's 10 Europeans, North Americans, South Americans, Asians,
Yeah.
and 20 Africans.
Yeah. Because of course it's theoretical neuroscience is, of course, it you need computers, or at least access to computers and internet and stuff, but it's even I mean, obviously, like in South Africa you also have strong experimental labs, but not everywhere in Africa I guess they have this strong experimental activity. But there's in the computation neuroscience, if you are well hooked up and use more of these sort of like resources, if you're and yeah, so this is sort of like a nice, if you think of all these people who have access to internet now, people
Yeah.
sitting in
Yeah, it's the
cafes.
same idea, right? Like it's the same idea, just sort of giving it a starting point in person, and then hoping
Yeah.
that the internet will be able to provide
Okay.
enough
So
of an
this is
environment to carry the torch onwards.
so I think it would be really cool if you could also mobilize brains in very populous countries which maybe have less sort of scientific experience, like I don't know, Nigeria, Indonesia,
Yeah,
and
exactly. Nigeria,
like
South Africa, Ethiopia,
huge
Tunisia,
countries,
Egypt.
which are huge populations, and
Yeah,
so that would be really cool. It takes a world. What do they say? It takes a world to understand the brain. Isn't that what they say? So I think
yeah, maybe.
that's
I'm
true.
I'm sure it's true.
I think it's probably several worlds to properly understand it, but
Yeah,
good.
yeah.
Okay, well, thanks a lot, Tim. It was very nice to have you here, and thanks for
Lovely hanging out.
yeah. Good luck. No, good luck. What is?
Good night and good luck.
It's actually good night. Yeah, it's a bit late here. Actually, it's 9. No, it's 11. 10:40 in the night. So So that's why. Good. So okay.
See you soon. Thanks.
Yes.
Thanks for having me.